Welcome to Brain Ponderings, your window into the world of neuroscience research. Conversations with scientists at the forefront of their particular area, the large neuroscience field. And today it's my great pleasure to welcome Shuying Sun, sorry. She is Associate Professor of Physiology, Pharmacology, and Therapeutics at Johns Hopkins University School of Medicine. She's also has appointments in the Brain Science Institute and the Department of Neuroscience. She is an expert on RNA in the brain and in relation particularly to abnormalities in RNA in neurodegenerative disorders. This is a topic that it, it was kind of ignored for a long time in the field of neurodegenerative disorders because everybody was focused on the proteins themselves that were forming aggregates out of neurons and what's causing the protein to whatever misfold self-aggregate after it's formed. But now there's some genetic and experimental evidence that I will talk with her about. So welcome,
Thank
Shuyi.
you. Thank you for having me here.
Can you talk about your background? So you grew up in China
Yeah,
and did
yeah,
your undergraduate there?
right. Yeah, I finished my undergraduate education and then came to US for PhD study. So I actually joined the graduate program in the New York State University at Stony Brook. But then the program had a partnership with Cold Spring Harbor, so I did my PhD thesis in Dr. Adrian Krenner's lab at Cold Spring Harbor laboratory, which is really the founder for molecular biology. So I get training on RNA splicing, the basic molecular mechanism of RNA binding proteins in Adrian's lab.
And so, I mean, everyone listening to this will know that, that you have genetic code in DNA and it's transcribed into RNA, single-stranded RNA, and then that happens in the nucleus, and then the RNA gets spit out of the nucleus. Actually, it doesn't really get spit, but there's actually elegant mechanisms. And then when it gets typically in the cytoplasm, or actually endoplasmic reticulum too, then that RNA sequence is translated into amino acid sequence. So can you talk about what's happening in the nucleus kind of specifically and how the RNA gets out?
Yeah, sure. As you bring up, so it's like the genetic flow is from DNA, RNA, protein, right? So of course, people think the protein is mostly the final functional molecule, right? But RNA is at the same time is not just a simple intermediate, but they're actually highly sophisticatedly regulated to maintain the right amount of gene expression, right? So after transcription, to have a pre-mRNA, and then this need to be processed, like 5' need to have a M7G cap, and then 3' end have the polyadenylation tail, and then the introns in the middle have to be retained, removed properly so that the exons are joined together to form the mature messenger RNA. And then only the fully processed mRNA get exported from the nucleus to the cytoplasm, which is not just a random event, right? It's a protein complex recognized the fully processed mRNA actively exported to cytoplasm, where translation happens to produce the proteins. And
There
so,
must be some evolutionary reason why this, you have initially the pre-mRNA and then it's whatever spliced into smaller bits and they're patched together into the final. It seems inefficient, but is there a reason why
yeah,
it actually is efficient?
I guess actually during evolution is actually become the splicing is actually become more and more complicated. The intron get bigger, right? Actually, exons are the smaller piece of the DNA, and then But by doing this, I guess one benefit is like from each gene, you can produce multiple isoforms, right? By using different combination of exons or different combination of splice sites. And then on one hand, this can, as you can imagine, it can generate many different protein isoforms from one gene, right? These different proteins could have different functions, different localizations, right? But also, it's also a mechanism to, I think it's fine-tune the gene expression level too, because some of the isoforms are not productive. For example, for the isoforms that have a premature stop codon, basically, if you have a stop codon that is before the last exon, this message is recognized as kind of an error during translation. So those message RNA should be degraded. So if you're changing the isoform ratio, even though the transcription level might be the same, but if you produce more of the non-productive isoform, you have less protein as well.
Yeah, so it's actually more efficient this way, and increases the diversity of
Right,
of,
right.
of both regulation and the actual final protein product.
Right.
Yeah,
Right.
cool. And then how do these RNAs get out of the nucleus? So the nucleus has two membranes. It's
Right.
a
It has a
double
nuclear
membrane.
pore complex, right? So that's the place where molecules travel in and out. For messenger RNAs, usually when after splicing, there will be a EJC complex deposited near upstream of each exon-exon junction, and this protein also interact with like other RNA binding proteins, and then interact with the NXF and X1 complex. Those one can mediate going through the nuclear pore, carrying the RNA and the RNA binding proteins together. So it's not to make the RNA get out.
So these pores are pretty big. They have to be pretty compared,
Yeah.
like I think a lot about ion channels in the the outer membrane of neuron, and they're small. So how big? So these proteins can go through them.
I think for protein, right? Protein synthesized in the cytoplasm, right? But the nuclear proteins need to go in, right? It's also through nuclear pore. So if you think about protein size, I think for proteins below around maybe 30 or 40 kilodalton, those can freely diffuse in. So you don't need extra helper to transport. For the bigger proteins, you need like importing those kind of machinery to help import, which, it should be bigger, right? Because you have a complex to go in. So I think it's allowed, but then it's not just like molecules can just go in and out. There are regulations about this process.
Yeah, but beginning when I was a postdoc, and then when I started my faculty position, so this is in the 1980s, and Roger Chen just came out with the fluorescent probes. He developed these fluorescent probes for calcium that you can introduce into living neurons, and you can kind of image under the microscope relative amounts of calcium. And one thing I found in the hippocampal neurons was like I'd load the cells with the calcium indicator dye, then I'd get a big calcium signal indicating high calcium levels in the nucleus. And there's some evidence that somehow, even though you got these big pores in the membrane of the nucleus, you can have ion gradients, which it doesn't doesn't make sense, right? I mean, because if they're big pores, you'd think that.
Right.
So I think there's still a lot of work that needs to be done on. I think there's some regulation there, even though you can get the proteins diffusing, somehow there's more regulation than that.
Yeah. I think it's definitely a very important topic also in neurodegeneration because, like besides protein aggregation, those RNA binding protein mislocalization is also a very important feature for neurodegeneration because when the protein is localized to the wrong place, it cannot catalyze their normal function.
Yeah.
And the nuclear pore, some proteins in nuclear pore is one of the like most long-lived protein. so I think there are a lot of interest in the aging and neurodegeneration, trying to understand the nuclear pore or nuclear cytoplasmic trafficking defects.
Now, there's one neurodegenerative disorder in particular that seemed to be at least my reading of the literature, the beginning of a lot of focus on RNA and neurodegenerative disorders, and that's ALS, motor neuron disease. Can you talk about these two proteins that have to do with RNA and are related to ALS?
Sure. So I guess that's the reason also for me like to do my postdoc in ALS field, like in Don Cleveland lab in UC San Diego. It's because I mean for sure from the basic RNA biology we know they are most complicated RNA processing in the nervous system, right? And then I think it's in about 2006 that's when TDP-43, which is the RNA binding protein, was first found to be a big component of the protein aggregation in ALS FTD.
Yeah.
So this is beginning to bring attention to the field that maybe RNA binding protein or RNA processing is also important in neurodegeneration besides protein part. And then in 2008, there are genetic mutations of TDP-43, I think also fast, which is another RNA binding protein, are found to account for about 5% of the inherited ALS patients. But then the aggregation part is actually more profound because that's a protein pathology of TDP-43 is found in more than 95% of all the ALS patients, including inherited and sporadic, and half of the FTD patients. And now it's also found in about half of the Alzheimer's disease patients. So
my father had dementia, and it's an interesting story because I grew up in Rochester, Minnesota, where the Mayo Clinic is, and one of the kind of very prominent person in the clinical area of Alzheimer's is Ron Peterson, who's there at the Mayo Clinic, and right. So my dad's short-term memory getting bad, and he was diagnosed by Ron Peterson with probable Alzheimer's disease. But then when he passed away and the autopsy was done on his brain, it was actually, he didn't have a lot of amyloid accumulation. He had some, but it wasn't reached the criteria for Alzheimer's, but he had quite a bit of TDP-43 pathology. And he had massive loss of neurons in the hippocampus, some in the frontal cortex, and it's some tau pathology, but the main thing was TDP-43. So, so it's very important, not just for ALS and frontotemporal dementia, but I think also for...
quite a broad neurodegeneration disease.
Yeah. So this TDP-43, can you talk more about that? So where is it normally located in the neuron, and what happens? Where does it go in ALS? And you mentioned loss of function. Is there any gain of function?
Yeah, definitely. So normally it's predominantly localized in nucleus, but also it is a shuttling protein as well. So it can shuttle between nucleus and the cytoplasm. Because I think many RNA binding proteins have multiple functions in different steps of RNA processing. So of course, in the nucleus, there are many different RNA processing steps, but then they are also need to be regulated in the cytosol. I guess especially important for neurons as well, because some RNA definitely traffic into very distal regions, right, like dendrites, axons. So which I also could have a localized translation, which might be very important for neuroplasticity. So many RNA binding proteins do have many different functions. For TDP-43, definitely it's mostly localized in the nucleus. And one major function in recent years that is well characterized is that they can regulate alternative splicing, especially it's a repressor of cryptic splicing. Basically, those basically it's like there are potential splice sites buried in the introns as well. But then when TDP43 bind there, it's kind of a block the recognition of this splice site. But if you lose the function of TDP43 in the nucleus, then now this splice site can be recognized by the spliceosome and include a short piece of intron that should not be present in the mature mRNA. Then this abnormal splicing could alter the gene expression of many neuronal function-related genes, changing, as I said, probably many of these, because the included extra exon is out of frame, so it introduced this premature stop codon. Therefore, the full-length transcript is reduced, that it cannot produce enough of the normal proteins. And there are also cases that if it's in the frame, it could produce a protein that have extra domains or something.
So in ALS, then the more of TDP43 is outside of the nucleus than normal.
Right. So for pathology, right, the most obvious phenotype is the aggregation, right? And the aggregation is mostly in the cytoplasm. But there could be also evidence that there could be loss of function or mislocalization even before the aggregation. In a way, I guess these two process, aggregation versus mislocalization, could be like synergistic, right? When you have more mislocalization in the cytosol, you have more prone to form more aggregation. Of course, the aggregation could also trigger some like toxicity because of the aggregates part, or it disrupts the protein homeostasis pathway, which could be a gain of toxicity. So that's why TDP43, maybe the mechanism for disease is kind of complicated. So it's definitely have a loss of function part, which is increasingly believed to be very important, but there might still also have this gain of toxicity part.
Shuying, has someone developed a way to have a cell, a neuron, with TDP40 aggregates in the cytoplasm, but in the nucleus, TDP43 is functioning normally?
Yes, so this is so far the most of the model we can get that for example, traditionally, right, when we find a disease mutation, like we build a mouse model to study, right, the mechanism. And the most straightforward is to do a transgenic mice overexpressing this mutant gene, right? Yeah. Because because it is a dominantly inherited mutation in most of cases, right? You would think it's more like a gain of toxicity rather than loss of function, right? And this is basically most of the mouse model in neurodegeneration field, right? Like SOD1 for ALS, tau, A beta, right? I mean, Huntington's disease usually is overexpressing a transgene. So that's more mimicking the aggregation part. But the problem is for iron binding protein, basically the right amount of level is very important. So too low is not good, but too high is also not good, right? When you overexpress, even when you overexpress a wild type protein, it's also toxic. So it's not really a mutant dependent phenotype.
I guess what I'm asking, can you overexpress it and not have the overexpressed protein in the nucleus?
Well, I mean, people have done that. You mutate the nuclear localization signal of the protein. So you overexpress the protein in the cytoplasm.
Yeah. And then is that sufficient to cause degeneration?
That can cause degeneration, but the question is whether it's really representing the whole picture for the disease. Because you would hope that when you have a cytosol aggregation from a transgene, it's best if it can also sequester away the endogenous protein, right, away from nucleus. Then you have both gain and loss. But so far, we don't have this model. So the endogenous protein is still there. It's still functional. So you can either have loss by knocking down or aggregates by overexpress, but there's no model can have both that can faithfully mimicking the human disease condition. So that's quite one challenging. And also one challenging when you design therapy, right? So for many neurodegeneration disease, one therapy is to just knocking down this toxic gene, right? But for TDP, obviously it couldn't work because you will make it worse because you lead to more loss of function.
Yeah.
Yeah.
What was your main finding in San Diego and Don Cleveland's lab?
At that time, I mostly focused on like I started looking for mechanism underlying the FAS, which is another RNA binding protein, and then I also like for SOD1 mice, I explored what could be the cell type specific gene profile changes that underlying maybe a cell type specific vulnerability for neurodegeneration, and then in the middle of my postdoc, there's new discovery of the C9 of 72 repeat expansion. So I also begin to get involved in the C9 case. I kind of together with other postdocs, I discovered like, for example, there's an antisense repeat produced from that locus besides the normal sense transcription.
Okay, and so then you decided when you started your own lab, you're going to focus a lot on the RNA processing and the nitty-gritty of what's going on there. Is that right?
Yeah. So basically, this is what I mean. Our major focus trying to understand the dysfunction of RNA processing, how it contributes to the disease mechanism, and I think when in the beginning of the lab, we really focus a lot on C9 or F72 repeat expansion, and gradually also expand to RNA binding proteins like TDP43.
I see. So can you talk about then the C9 or 72
Sure.
and what's happening with
Yeah.
them?
So this is a disease mutation that is one of the repeat expansion example, right? So it's not caused by a single nucleotide mutation like many other like TDP43 or FAS or SOD1, but so this is a repeats of microsatellite repeats, which usually 3 to 6 nucleotide, and this normally actually exists in the human genome, right? It can potentially playing regulatory roles, but then this repeat is also kind of unstable, have a tendency to expand during.
Right. So in Huntington's disease, you have a repeat expansion polyglutamine, right? In the protein, you get
Right.
a
Right.
string of excess
Right.
glutamines.
AG repeats.
Now, but with the C9orf72,
So it's a hexanucleotide repeats. It's GGGGCC. 4 G's, 2 C's.
yeah,
And it's located in the intron. So not influence the protein coding.
right,
Normally you have should have less than 10 repeats.
yeah,
But in the patient expanded to hundreds or even thousands of repeats. So it's very long. Usually, I have to say, usually repeat expansion in the non-coding region is longer than the repeats in the protein coding region.
So then this repeat is kind of increasing the distance between the parts of the mRNA that are going to be translated into protein after all the splicing. Is that true?
Well, I mean, it's in the intron, so it's in the piece that is removed. So for the mRNA, it didn't change sequences at all. So it's really just, I guess it's during a co-transcriptional RNA processing, it makes the splice site more distal, right, more far away.
Right.
So there are definitely kind of evidence suggesting it partially inhibits the splicing efficiency, but it's not that big. It's not just completely block it. So there are multiple hypotheses of the disease mechanism caused by this G4C2 repeats because, first of all, there are models that this repeat RNA, I mean, it's true, I mean, it's validated that this repeat RNA can form nuclear foci or granules-like structures because it's it's a repeat sequence, 100% GC rich, so it's resistant to the nucleus digestion. Because normally introns after splice out, it should be degraded, right? But then this repeat cannot. So if you do a RNA fish, you can see those kind of puncta in the nucleus. And those nuclear RNA foci can potentially disrupt RNA binding protein functions. And this is this is not the first repeat expansion disease, right? There are many others, like I think, and for the repeat expansion located in the non-coding region, there are RNA-mediated toxicity, right?' of the biggest, well-studied case is about myotonic dystrophy. They have a CUG repeat in the 3' UTR, and then that repeat RNA foci specifically sequester RNA binding protein that leads to the loss of function of that RNA binding protein. So this is the one scenario, right? But at the same time, this repeat RNA can also be translated. So from a very non-canonical mechanism, right? So it does not have AUG start codon and does not have 5' cap. But the translations can still happen and in all the possible 3 reading frames. Usually, for normal transcript, you only use 1 reading frame, right? Because you start from
it's then you get a 2 amino acid repeat?
Yes. Yeah, that's exactly.
Yeah.
You get the dipeptide repeats in the different reading frames.
And what what amino acids?
So it's from the sense G4C2 repeats is GP, GR, and GA.
Okay.
The 3 different kinds from 3 different reading frames. Okay. From antisense, which is C4G2 repeats, you have PA, PR, and GP again.
Huh. And do these and then those accumulate in the
you can find if you use antibody, right, for specific dipeptide repeat protein, you can find aggregates in the patients.
And are those aggregates occurring just in motor neurons or all over? Are they all neurons, not other types of cells?
Yeah, it's a good point. So first of all, the C9 repeat expansion is actually a shared genetic cause for ALS and FTD. Yeah. So it's not only motor neuron, but also for other types of neurons especially for FTD case.
And what about other types of cells?
Right. I think there are also evidence like in astrocyte, for example, but I don't think there are a lot of systematic study for the other cell types.
And so one way that these dipeptide repeats and this expansion of the introns could affect the cells is by altering gene expression, I guess, or RNA binding proteins or affecting that. So have people done transcriptomics, proteomics, and kind of is anything clear come out of that?
Yeah, so definitely there are a lot of studies doing the transcriptomics or look for RNA binding proteins that can be sequestered by this repeat and linked to the function of these RNA binding proteins. And I think, well, there are many publications talking about different kind of RNA binding proteins. I guess maybe one difference from the CUG repeats in myotonic dystrophy, I feel, is not because in that repeat there's very specific to one of the RNA binding proteins very predominantly sequestered and leads to loss of function. For C9, I think we don't really have that unique one that can explain everything. But instead, we actually recently think a different way, that we think that actually this might influence RNP granule dynamics, which is formed by liquid-liquid phase separation, right? It's important for many different RNA processing steps. So what we found is actually this G4C2 repeat RNA can localize to nucleus speckle, which is a compartment important for co-transcriptional RNA processing, especially splicing. So we do see that there is a trend of increased intron retention and exon skipping in the C9 patients. So kind of suggesting instead of influence a specific one RNA binding protein, it could also influence the general splicing machinery and reduce the overall splicing efficiency in the whole transcriptome.
Okay. So there's several of your papers that really attracted me to your work. One has to do, and maybe you can just kind of summarize these. They're all in top journals. So you did one where the C9orf72 repeat is upregulated by stress, cell stress. So if you expose cells to stress.
Yeah. So this is one of our early work. So basically, I mentioned this repeat has very non-canonical translation, right? Does not need the AUG, does not need the required 5' cap. And for canonical translation, the translation efficiency mostly influenced at the initiation step, right? So you need initiation factors, protein factors that can bind to the 5' end, 5' cap, and then begin to scan on the mRNA from 5' to 3', and then until it found the first AUG start codon, and then recruit other like ribosome to form the ATS ribosome to start the translation elongation to make proteins. So there is a stress signaling from the cell that's called the integrated stress response.
Yeah.
It's a mechanism for cells to help cells to adapt to a stress condition, right? So when there are different stress stimuli, for example, oxidative stress, accumulation of the misfolded protein, right? ER stress, or maybe inflammation. So there are different kinases get activated, and they all come to a central regulator that is EF2 alpha. And phosphorylation of EF2 alpha will lead to a global translation shutdown.
Yeah.
But there are also exceptions. A subset of mRNA, their translation is actually upregulated, and those translation usually is cap independent translation. So when we study the repeat translation, which is also cap independent translation, we think, well, how will stress influence this kind of non-canonical repeat translations? Because stress response, this integrated stress response pathway is very relevant to neurodegeneration diseases, right? It's found to be elevated in many disease conditions.
And also, even though these genetic cases of ALS or Alzheimer's or Parkinson's, usually they have an earlier age of onset, but nevertheless, you don't see the pathology or the dysfunction until the individuals are at least, say, in their 40s. And one thing that's changing, even during that early period of aging, is that as you get older, the ability of your cells to adapt to stress becomes impaired. And so when I saw this paper, I thought, oh, this may be an explanation for why the age association, why aging is a major risk factor for onset, even in the genetic form.
Yeah, I definitely. Because acute stress response for this signaling pathway is meant to be protective, but if it's a chronic stress accumulation, it will be harmful for the cells.
Yeah.
And for this specific case, it's just because there's also repeat expansion. So basically, you even accelerate production of more of the toxic proteins, rather than the toxic proteins also increase the stress level to the cells, right? It's like a forward feedback loop, right?
Yeah.
And kind of accelerate to accumulate more of toxic things and contribute to the kind of maybe cell degeneration.
maybe even related to that is a very recent paper you had in Neuron where you have this, I think it's phosphatase is influencing the degenerative process in C9orf72 ALS.
Yeah, that's kind of a
PTP alpha. Yeah.
PPP, yeah, sigma. So this is
Yes.
actually we start from because our lab also do a lot of CRISPR screening to identify genetic modifiers, and for that particular screening, we basically start from toxicity from one particular dipeptide, poly-GR, which has been shown to be the most toxic dipeptide. So we know, okay, when we overexpress this dipeptide, it will gradually cause a neuron death.
Yeah.
And we did a CRISPR screening and to look for genetic modifiers that which one can, when loss of function, can protect the neuron from that GR-induced cell death, basically make a neuron more resistant to this toxic protein. And this is one of the hits we followed, and turns out to be a PKD signal, which is a phosphatase that can influence the PI3P level, and which is very important for vesicles like endosome, lysosome,
Yeah.
autophagy,
Yeah.
which is also a very important aspect for neurodegeneration disease. That's
Right.
a little bit go beyond
So,
RNA processing, I would say, but it's very important for like understanding the biology of neurodegeneration.
yeah. And right. But in that paper, you also, so you use the CRISPR screen to identify that phosphatase. And then there are specific drugs, I guess, that inhibit that phosphatase. And you found that if you treat mice with this dipeptide repeat that have the pathology with this phosphatase inhibitor, then that is beneficial and lessening the pathology and improving the function, or at least, yeah, improving the motor function, I guess, which is kind of a behavioral readout in
Yeah,
these mice.
yeah, yeah.
Yeah, so that's neat. I mean, you go all the way from a CRISPR screen to showing that if you inhibit the target that you found, that's beneficial.
Right, right.
So that's like a preclinical study that suggests the potential translation to
Yeah, I think
not translation of mRNA to protein, but translation of mouse studies to
so.
human ALS
Yeah, yeah.
patient.
Right, right. Yeah, yeah. We definitely think it's a like CRISPR screening is really a very powerful tool, right? To really allow you to like unbiasedly to look for new genetic modifiers, and you can go from very upstream mechanism study and try to understand or find the target and try to understand how it worked, and then because for this particular case we actually use a kind of so-called druggable library, so basically contain genes that they are already inhibitors developed, either it's
Yeah.
already FDA approved drug or in the as a research tool, right? They are already kind of a compound or different type of inhibitors available. So once you found a gene, you can quickly test these candidates inhibitors for their efficacy. And of course, usually one challenge is that many of, for example, the drugs developed for cancer didn't really pass the blood-brain barrier, right? So, but in this particular case, it's actually a reagent that developed for spinal cord injury, so it can pass blood-brain barrier. So it's definitely easier for us to test the efficacy in the nervous system.
What's the inhibitor? Do you remember what's the name of the inhibitor? I can't remember from the.
Actually it's a peptide ISP.
Yeah, oh, I know. Yeah. Because we did some work on calcium-dependent proteases, and uh, but also on protein phosphorylation and Yeah, so this kind of background here, some people may know detail what we're talking about, others not. So the function of proteins can be regulated by either the adding or removal of phosphate groups to certain amino acids on the proteins. Usually it's serine, threonine, or tyrosine. Actually, those are the three main amino acids. So there's a whole bunch of enzymes called kinases that add the phosphate group and then phosphatases that remove it. And this is the way, neurons, this is studied a lot looking at like uh, it's post-transcriptional regulation, essentially. You can get quickly change the function or relative level of function of a protein, for example, by causing calcium influx to the cell, which, activates kinases or proteases. And so this is the way in neurons where, as you mentioned, you have long axons often extending even millimeters or more centimeters and elaborate dendrites, and they're way away from the nucleus. And it's very important for the control of of synaptic function and neural network function to have these local changes. So you get activation of one synapse, you get changes in that synapse, but not another. And often these are mediated by these mechanisms. So I think this phosphatase sigma, PTP sigma, I got to read up on it because I'm not sure, because this could suggest that these so the poly-GR, these dipeptides are getting out of they're in the cytoplasm.
Yes, yes.
Right. So in theory,
So for this,
yeah.
when we express basically when we express this poly-GR in neurons, it's mostly diffused in the in the cytoplasm. And actually, when we knocking down PTP sigma, which can protect the neuron, we actually when we do the Western, we do not see the GR protein level change.
Okay.
So it's not the
Okay.
mechanism not through reducing the
Yeah.
toxic
All
protein
right.
level. It just makes the neuron more resistant.
Yeah.
But on the other hand, later when we test mouse model that can have GR aggregates, we do see the aggregation gets reduced. So I think it might relate to the lysosome autophagy function that can promote the clearance of aggregate form.
Okay. In that case, would you predict that in a TDP-43 model, if you inhibit this same phosphatase, it would also be beneficial?
Yeah, this actually we are testing that exactly what we hope
Okay.
to do.
Okay. Right. So this could be a general approach.
Right, right.
Huh. Okay. That's very promising.
Yeah, we are testing, hopeful to have a positive answer.
Yeah. Yeah. Okay. Then what, what's you have several other really kind of, it gets into areas I don't know some, a lot of details about, but you're doing, for example, you had this one that's in preprint form on these this retrotransposon RNAs, Lin1 retrotransposon RNAs, and um, you're looking at, I guess, interaction between those and the C9orf72 and TDP-43. I know this is something so Rusty Gage is a friend of mine, and I know he was doing something with these Lin1 RNAs, which are kind of fascinating. So every neuron, so idea, I guess, is every neuron in the brain, well, we know every neuron in the brain is different, but um, there's during the, right, I think I got this right. During the lifetime of a neuron, you can get changes in these that have important roles in, for example, selective neuronal vulnerability or something like that.
Yeah, I think actually we come to the retrotransposon line 1 RNA is really uh, because we have a previous work studying M6A RNA modification that
So these are similar. So there was a Nobel Prize a long time ago given to, was it McClintock, for work in plants on
for which one? Oh yeah, oh, yeah,
transposons.
yeah, yeah,
Yeah,
for transposon. Oh yes, yes,
yeah,
in
yeah.
the clones, right?
So they're
But we are
jumping
yeah,
genes called jumping genes or
right,
something.
right. So this is, in mammalian cell, right? It's mostly active in stem cell, right? It's jumping around. So basically, it's a piece of RNA after transcribed. It actually should be translated to make two major proteins that can then bring the RNA back to the nucleus, then do the reverse transcription, and then insert to a random location of the genome, right? So it basically can start from one location and insert another copy to a new location. So
Yeah, yeah,
that's called jumping, right?
yeah.
And it's most active in stem cell stage and should be silenced in somatic cells. And I guess Dr. Gage's work showing that actually in neurons, it seems to be also one of the somatic cells that can have a higher line 1, especially then it's also known that during aging, the line 1 RNA is increased. And then it's also increased in neurodegeneration diseases. So there have been a lot of, I guess, interest to see whether, the jumping, whether it can maybe, for example, increase the DNA instability, right, DNA damage, therefore also contribute, one of the mechanisms contribute to neurodegeneration. So this is really from, well, when we study, we are not really specialized in retrotransposon, right?
Yeah,
We
yeah.
study RNA biology. The reason we come to this is because when we study the abnormality of M6A RNA modification, we published that it influenced the mRNA stability, influenced the C9 repeat RNA stability as well. We also noticed that it's also influenced the chromatin-associated RNA. And among the chromatin-associated RNAs, the most influenced one is a repeat RNA, that is line 1. That's how we come to here, because we know line 1 is abnormally elevated in neurodegeneration, and including ALS FTD, right? And now we also maybe find a new mechanism why this line 1 RNA is elevated. Maybe it's not, so basically we think this is not coming from the transcription part, right? It's coming from maybe the RNA stability could also contribute
I
to the
see.
line 1 RNA accumulation during aging or in the specific disease context. So in this, for the C9 part, is because in the previous work we showed that there's an abnormal reduction of M6A level in the patient neurons. And then M6A can function as a degradation signal for degrading the RNA. So it should be the nucleus, or in this particular case, in the nuclear RNA, we found it's a nucleic exosome complexes that can degrade the M6A marked RNA mediated by a M6A reader protein, right? So, but if there is a loss of M6A in the C9 patient, then those RNA are less degraded, so accumulated more.
Huh. Yeah.
And in the TDP-43 case, the connection is because that the TDP is known to bind to these repeat RNAs, retrotransposon RNA. But we show that TDP actually interact with this nucleic exosome. So basically, they can bind and then mediate this target to the degradation machinery. But if you don't have TDP, this target can also not be degraded anymore. So therefore, also increase the accumulation of this line 1 RNA.
A long-standing, general question in neurodegenerative disorders is that of the selective vulnerability. That is, why are motor neurons the neurons that degenerate first and most in ALS, and why, say, hippocampal neurons in Alzheimer's, or uh, medium spiny neurons in Huntington's disease and the striatum are dopaminergic neurons in Parkinson's. And Do you think your work can help address this? So there has to be something going on that's different in motor neurons that makes them more vulnerable to the C9orf72 and the dinucleotide, or not, dipeptide repeat than other neurons.
Well, for C9, I think it's, well, I do think C9 is not only motor neuron, but of, I mean,
No. I mean, but that's the case for all of these neurodegenerative disorders that,
right.
the
So,
mutations in amyloid precursor protein aren't just in hippocampal neurons, they're all over, or mutations in Parkin or
yeah.
alpha-synuclein. So, um, one thinking, we actually did something relevant to this way back when I was at Kentucky, uh, and so that was when the superoxide dismutase 1 mutant mutation was discovered and the first mice were generated. And we were doing this work where we found that intermittent fasting was neuroprotective and caloric restriction in animal models of Alzheimer's, Parkinson's, stroke, even Huntington's disease, Huntington mutant mice. But then when we tried the SOD1 mice, the intermittent fasting, it not only wasn't beneficial in terms of delaying onset of motor dysfunction and degeneration, but once the mice started to show symptoms, they went downhill quicker and died more quickly. So it's the only neurodegenerative disorder where anti-aging, so caloric restriction, intermittent fasting are the only ways we know we can reliably counteract aging and extend lifespan in normal animals. And it's very good in suppressing a lot of disease processes. So then I did some reading and it's very interesting. So it may be that the motor neurons are not, the intermittent fasting is actually a stress on cells. And that's why it's good. It's a good stress because the cells respond adaptively by upregulating uh, stress resistance proteins, for example, heat shock proteins.
mm-hmm.
And we found that others actually, that motor neurons don't show heat shock response like neurons in other regions of the nervous system. So, um, one potential reason is that these neurons, for whatever reasons, aren't good at responding to certain types of stress, maybe metabolic stress. Um, so I don't know. I just throw that out there. You did this little bit with stress responses. And so this question of selective vulnerability has not been answered. So
No.
if you could answer that, you'll get well, I don't know if you get the Nobel Prize, that's hard, but you'll get my prize. I'll give you a prize.
Yeah, I guess it's also definitely it's a very important key question, but I guess different specific genes might have slightly different. For example, C9. I mean, and I think there are multiple layers of contribution. For
Yeah.
C9, for example, well, the expression of C9 gene itself, it is high in neurons than like astrocyte, than all your skin fibroblasts. So there could be one contributor that because it will express more than
Okay.
other cell types, right? In
Yeah.
this specific case. And of course, there could be others. For example, when you like deal with misfolded protein accumulation,
Yeah.
motor neurons definitely, I think, have some different properties than others.
Yeah,
Right?
and
They
that's
are set
related to the
on.
heat shock.
Right.
These protein chaperones are very important.
Right.
Yeah.
So, for example, when you have a similar amount of accumulation, other cells can still deal with it, but motor neuron cannot.
Yeah. Yeah.
I think maybe because, for example, it's in a way it's like a gene network, right? So for a specific function for motor neuron, you need this kind of a amount of different genes or pathways, but then it's not good enough to deal with some kind of stress. That
Yeah.
might be one reason that it's become more vulnerable under certain conditions than other cell types.
Yeah. And then another question related to that is, so for sporadic ALS, it's not very common, right? I guess I don't know exactly, 1 in 10,000. I don't know, but anyway, it's not real common. It's common, not nearly as common as Alzheimer's or Parkinson's, for example.
Right.
And so then the question is, for the, why do some people get ALS and others don't? And is there any role for alterations in RNA processing, RNA binding proteins, uh, you know, comparing people of the same age who don't have ALS to those who, I guess ideally you want to be able to go, we can't though, is be able to know who's going to, I guess you want to do somehow a longitudinal study, something like that.
Agree, but that's so challenging for brain disease, right? So that's
Yeah,
exactly what is missing or difficult for studying the disease mechanism for neurodegeneration disease,
yeah,
because what we see from humans is out in the end stage, right?
yeah,
That's why many, previous pathologies appeared about protein aggregation, right? But you even don't know whether the aggregation is really a driving force
yeah.
or it's a consequence of cell death, right? So
Yeah,
really,
you can see a little bit of what's happening, but you have no idea.
right. So it's important to have a longitudinal study, but
Yeah,
not so easy. I mean, that's why I think for therapeutic development, there are a lot of emphasis on biomarkers as well, trying to do like
yeah.
CSF or even better to have a blood, right? Biomarkers that can kind of predict your progression, so that it can also have a better treatment.
Yeah, there's a lot of work in Alzheimer's field on that now, having blood-based biomarkers and then that, and even the clinical trials are moving towards that. And the reason is that the classic way is to look at the clinical symptom, the memory loss in Alzheimer's or the motor weakness, in strength weakness in ALS and the progression. ALS progresses pretty fast, but Alzheimer's progresses slow. And any individual, there's fluctuations in their performance on the memory test. So someone who's getting, say, early stages of Alzheimer's on today, they may not do very well. And then you come back and test them 6 months later and they do a little better. Even though if you look, over 5 years, there's a clear decline, but it's kind of like, this. And it's really hard to get any good or clear results from the clinical trials unless there's some intervention that is like reversing the damage.
Yeah,
Yeah, that's a big
yeah,
problem.
yeah, exactly. That's why I
But
think a lot
but
of
if you can have a biomarker that
right.
reports essentially reporting how many neurons have died in the last whatever month, and you can distinguish differences, then that could be useful. And
Yeah, yeah.
and yeah. Okay, what's your most exciting? So how big's your lab now, Shuying?
Around maybe like 12, 13 each people. Yeah.
Ah, okay, that's good. That keeps you busy.
Yeah, yeah, yeah.
Mostly postdocs, or
Actually half now, like
yeah, okay.
grad student and postdoc. Yeah.
Yeah, yeah. That's good. Don't get too big.
Yeah, yeah.
Don't get too big a lab, then all you'll be doing is sitting at your desk. Maybe you're already doing that all the time, but my lab got too big, I think, and yeah, then what happens is you spend all your time writing and whatever, writing grants and stuff, and then you spend less time actually with the people in the lab.
Yeah, I mean, I think it's a good size now. We still have a lot of interactions,
Yeah.
and then it's a good size for people to talk
Yeah,
to
I agree.
as well, right?
That's a good size.
Yeah, yeah. But it's, nowadays it's hard to get bigger with the current funding situation.
Yeah, that's true. That's true.
Yeah, but
Okay, what's your most exciting project you can talk about now?
I mean, I think we continue to, do different kind of CRISPR screening to help us identify more modifiers.
Yeah, yeah.
And we like to go deep after we focus on or validate one target, really trying to understand exactly how it works.
Yeah.
And then we also hope, like, I think we do a lot of basic research for mechanism, and then we start from maybe just the cell models, and for screening, you have to use a large scale. So, right, you can not directly use the patient material, right? You have to, and for mechanism, it's easy to just use regular cell lines or iPS neurons. But then after we get a better idea of the mechanism and show the effect, we always go back to the patient-derived neurons to test the disease relevance and the patient's postmortem tissues to see whether those pathways are also altered in the patient context. And then we also have like kind of an interesting hit. We would willing to work with like pharmaceutical companies to test more translational aspect, right? To design more pharmaceutical way to target those kind of gene or pathways to really proceed to the more preclinical study as well. So we do hope we have a lot of mechanism study, but we also hope to apply this mechanism insight into a more like therapeutic development.
Yeah, that's really you're in a really good situation and place to be able to do that. Not everyone is able to, to go from, discovery of new basic things to translation, and it's an exciting time.
Yeah. I'm still exploring that part. I definitely know less about that more translational part.
Yeah, but that's good.
Yeah.
Great. Okay, Shuying, thanks for taking the time for this, and have a good holiday season.
You too. Nice talking with you.
Yeah. Bye.
Bye.